A very good morning to one and all. Today's topic for the lecture is transfection techniques. Since we have learned about the genetic engineering, the basic steps involved in genetic engineering or cloning, the next occurs the transfection technique. What is transfection? Transfection is the art or the act of transferring foreign DNA material from any organism to another organism. Transfection could be of two types, stable or transient. What is stable transfection? Stable transfection is the type of transfection in which the foreign material which is to be transferred from an organism to another organism inculcates itself within the host DNA or the host genetic makeup and maintains its replication along with the host DNA. For example, if this DNA is to be transferred to organism A, then in what will happen in stable transfection? In stable transfection, this molecule will inculcate itself with the genetic makeup or the DNA molecule of the organism A, and as the replication of this molecule proceeds, the simultaneous replication of this molecule also proceeds. What happens in transient transfection? Transient transfection is the type of transfection in which the foreign DNA that is to be transferred from an organism to the host organism maintains its individual replication. That is, in this type of transfection, the foreign DNA does not inculcate itself within the molecule of the host and maintains its separate identity, that is, maintains its separate or individual replication. There are a number of methods through which transfection could be achieved. Transfection methods could be summed up into the following ways. Direct gene transfer and agrobacterium mediated transfer. This is also called as biological method of gene transfer. Then direct gene transfer is further categorized into chemical methods and physical methods. Chemical methods are of the following types: calcium phosphate mediated, dextran mediated, PEG mediated, and silicon fiber mediated. Physical methods are of the following types: electroporation, microinjection, particle gun, liquefaction, or laser induced. Whereas the biological methods could be of co-culture with explants or in planta transformation. Today we will study about the direct gene transfer, which are of the following two types. Chemical direct gene transfer could be done either by chemically or by physically. Chemical methods involves the use of certain chemicals such as calcium phosphate, dextran, silicon fiber, polyvinyl chloride, polyvinyl alcohol, and etc. First we will study about calcium phosphate mediated gene transfer. As the name suggests, calcium mediated transfer. In this method, we mediate the transfer of the foreign material into the host organism with the help of calcium and phosphate. What we do, we take a foreign cell we take the cell in which we have to transfer the DNA material, then we make a phosphate buffer and we drop the DNA particle which are to be transferred to the cell. Phosphate buffer containing DNA suspension. Then we take the calcium chloride solution and we straight away pour this into the phosphate buffer. When calcium chloride solution is poured into the phosphate buffer, what happens? Calcium and phosphate forms the complex and chlorine is liberated. So what occurs? A DNA gets coated with calcium phosphate. Now this calcium phosphate containing DNA is uptaken by the cell with the help of endocytosis. What happens when this preparation of DNA is made to come in contact with the cell in which these DNA particles are to be transferred? This cell uptakes these foreign DNA material with the help of endocytosis. Since we are using certain chemicals, that is calcium and phosphate, this method is enlisted in the heading chemical method, that is calcium phosphate mediated method. A calcium phosphate mediated method though have a very low efficiency, but this was the previously used method, earlier used method in the transfection technique. Then we move towards DEAE mediated. DEAE stands for diethyl amino ethyl dextran. In calcium phosphate mediated transfer, we were using the calcium chloride solution. In DEAE mediated transfer, we use the chemical called dextran. In this method also, the cell uptakes the foreign DNA material with the help of the process called phagocytosis. In PEG mediated transfer, PEG stands for polyethylene glycol. In polyethylene glycol mediated transfer, what happens is we put the plant protoplast in the suspension of the DNA and we treat that solution with the help of the PEG, that is polyethylene glycol. What polyethylene glycol maintains the pH of the solution or the cell into which the DNA is being transferred. Then comes the silicon fiber mediated transfer. As the name suggests, silicon fiber mediated transfer, here the transfer is taking place with the help of the certain fibers, that is silicon fibers. Silicon fibers ranges from 0.6 micrometer in diameter to 10 micrometer in length. We coat these fibers with the DNA and straight away transfer our DNA into the cell, that is the host organism. That it was all about the chemical methods. Then we move towards the physical methods. This was all about the chemical methods. We will study about physical and agrobacterium mediated transfer methods in the next class. Thank you.